Thank you. So I'll be talking about how we're using Sample Manager to manage our biomarker samples from the clinical sites where they're collected, all the way to the externally performed assays. So Kandel is an oncolytic viral therapy company. Our therapies target solid tumors, which are still a major cause of death and loss of quality of life. So some background on the mechanism of our therapies. Tumors can avoid destruction by the immune system because they can evade recognition through creating an immunosuppressive tumor microenvironment. There's therefore an opportunity to teach the immune system how to fight cancer in solid tumors. Our approach to immunotherapy centers on oncolytic viruses. They induce immunogenic cell death at the site of injection, which then unmasks tumor neoantigens with a now activated microenvironment. So this process has been shown to lead to a systemic immune response against the tumor, with evidence of clinical and biomarker activity demonstrated across several solid tumors. The feasibility and tolerability of our approach has been shown in over 700 patients across multiple phases of trials with our therapies. So we have two oncolytic viral candidates, an adenovirus platform and an HSV platform. We've been applying these candidates for different indications, as you'll see on this next slide. So CAN-2409, which is the adenovirus platform, has the majority of the clinical and safety data with trials in prostate, brain, lung, and pancreatic cancers. And then CAN-3110, our HSV platform, is being tested in high-grade glioma at this point. We have additional discovery programs ongoing with this platform as well that will lead to future indications. From each of these trials, we're collecting blood derivatives and tissue for research, looking at various immune biomarkers to better understand the mechanism of our therapeutics. So that's where Sample Manager comes in. We're managing all of these samples from all of those trials internally and had so far been using Excel spreadsheets for sample tracking. So when I joined the company in April, I started curating and improving the spreadsheets, knowing that eventually I'd import them into some kind of software. When using the Excel sheets, we, of course, wasted a lot of time asking around to find out who had the latest version or how exactly to interpret the data contained within them. And Excel doesn't log an audit trail, which is also not best practices when you're handling human biosamples. When we wanted to send samples out to a CRO for them to run, some of our assays, we were creating a separate spreadsheet and then saving that to a specific folder to indicate that a batch of samples was sent. So like here, you'll see an example of one of our curated spreadsheets that will be, imported into Sample Manager. So this has all the data that, we need to be capturing, which, I can look at and totally know what's going on, but isn't as readable to anyone else looking for some summary information. So I, would spend a lot of time making pivot tables and kind of summarizing the data from these spreadsheets for my colleagues. So in June, I started my search for a suitable product, and we were looking for something that could provide, of course, an audit trail for any sample manipulations by different users and would be searchable by anyone who, had access to the software. We also wanted to minimize the implementation time, and we didn't want to have to rely on workarounds or customization for too many of our core needs. So we did demos with a handful of companies and found that Sample Manager had both the software and the experience that most closely matched our needs. The other softwares we saw were more geared toward the tracking of lab processes that use a lot of automation or management of repositories of small molecules or biologics. And often, they'd require the software company's involvement in the implementation. But because LabKey Sample Manager comes from LabKey's other products, where they have this really extensive experience in supporting clinical trials like ours, it was a clear fit for us for our applications. And some of the features that really stood out as we were demoing were, this ability to track aliquots and how, each blood derivative can have, at least in our case, has many aliquots, like 5 to 10 aliquots. So being able to track those relationships was key. And also the way that Sample Manager handles different relationships between samples. I think being able to capture that information like from a source or a parent and bring that data into the grid of a derivative sample type really stood out to me. And when I asked, about that same feature from other companies, it sometimes sounded like more of a hassle, but with LabKey, there was a clear path, to how we could get it done, which was really reassuring. And I also, the dashboard, I think just adds a nice visual feature that some companies will like charge you to put together. And the straightforward bulk uploading I also, found pretty easy and makes it a lot easier to get our thousands of aliquots that we have into the database. And finally, because the software is such a good fit for our application, it just didn't feel like it had so many like extra features that would make it feel, bulky or difficult to navigate. So over the last month, I've been setting up all of our source and sample relationships, and I uploaded all the samples from our biggest trial through the bulk upload. And you'll see on the right here, a representative set of samples, the blood draw really being the focal point, because each blood draw comes from a patient who's enrolled in one of our studies at a clinical study site. And each patient will actually have blood drawn at multiple time points. So if you were to go up a level and look at the hierarchy of a single patient, you'd see this schema replicated here a few times, one for each draw. So from the blood draw, the site will immediately process the blood into plasma and Buffy coats, and then they also do the aliquoting immediately. So we decided to create these like virtual samples for blood draw, and they're processed right, just to help to connect the resulting aliquots to each other. So it's really these aliquots that are shipped to us, and we log them into the freezer, which they're now given a location in Sample Manager's freezer management tool, rather than sent out to our CROs or collaborators so that they can run the biomarker assays. These events are tracked as assays and logged out of the freezer, since the collaborator then depletes the contents. So the implementation and upload process for the samples so far has revealed to us some errors in our sample metadata that were contained in the Excel spreadsheets. And we found things like formatting inconsistencies, typos, location errors. We had like 2 samples that were assigned to the same location, all of which, as we were uploading the data, Sample Manager let me know that there were issues. So an example of this is this typo here on the right. I don't know how this happened, but somehow me or someone else typed an M and a G in the row in the column location. But when the error was thrown, I just could then find out where the sample actually was and fix it and register the sample. So it's also been nice, at team meetings, I've been able to quickly pull up the total number of samples or aliquots to report back to the team. And I look forward to getting more skilled with the filtering and searching. So as we begin to rely on Sample Manager even more, I think I'll be able to contribute, directly using the software during different team meetings. And I just wanted to highlight that, our implementation was a pretty quick process, thanks to both kind of the ease of use of the software, also, the really good documentation that's available, but also from the amazing support from Hannah and the team in, helping us work through all the different scenarios and how to represent our sample relationships. I spent just about 2 weeks setting up the sample structure and importing a few thousand aliquots. So that allowed our team to meet our milestones in regard to this project and also move on to other projects. I've been able to bulk register the majority of our samples that we have in-house, but we do have a bit more work to do before the implementation is totally complete. After finishing the registration of the remainder of the samples, the next step is for me to train my colleague who assists in receiving the samples on how to use the software. We'll then train some of our clinical research associates who have more interaction with the sites. And with them, we'll focus on, how they can update, access the information or update the entries as they receive more up-to-date clinical data. And I'll also add another sample type, which is tissue, which is going to be separate from blood draw, and we'll be adding our tissue samples there. And then finally, we want to add in some limited patient and assay data. But because, for each of our studies, we have kind of different sets of data that we want to capture, we have to think a bit about how, the best way to structure the data so that they can sort of all fit into one set of fields. So because of that, this is more of a longer term goal. But I'm definitely excited to continue forward with Sample Manager. And with that, I'll take any questions. Thank you.
Really great presentation, Jess. We'll give folks a couple minutes to ask some questions. In the meantime, I'm very interested in what you talked about and being able to share information at team meetings. How has that process been for you right now? Or do you just have Sample Manager up and just pulling things up as people ask questions, or do you have, are you using like a common grid or something?
Yeah, at this point, I'm mostly just like on my own personal screen, kind of keeping Sample Manager up so that when it's my turn to give a quick update on, how are things going with the samples, I can say, oh, we have, like 700 aliquots, or we received another blood draw this week. But I think, yeah, as I mentioned, like as I get more familiar with searching and kind of how I want to like structure these regular updates, I think that'll become something I can like easily export or, have something ready to go so that it's either accessible to others or something I can quickly pull up.
Awesome. And so we've got a few more questions here. Did you trial Sample Manager? And if so, how did it go? And did it help with your decision?
We actually didn't do a trial of Sample Manager. We spent a lot of time, on video calls with Hannah, looking into, some of the different things that we wanted to do with the implementation. And I think, yeah, once we saw Hannah kind of walking us through it, we were comfortable to go ahead.
And then another question, compared with higher end enterprise software, in your experience, did you find the procurement process easier or quicker?
I can give a little snippet on that, I guess. So maybe about a year ago at a previous company, I had demoed a handful of different software, and it took us like months to kind of describe what we were looking for and get the get quotes and kind of adjust the quotes to try to really better match our project. But in the case of Sample Manager, it was clear to us that it was a good match, and also the pricing was really clear. So we didn't have that sort of back and forth time, asking questions and kind of working on pricing. So I think for me, that turnaround time between, hey, I think I like this software, and we're actually implementing it, was really quick. So that was a huge benefit.
Awesome. Yeah, that's great to hear. And for other folks too, we have our pricing posted on our website. So, go to labkey.com and you can find it there too. Okay, thank you, Jess, so much. I really appreciate your time and presenting for us today.